So, hello. You have witnessed over the last 3 days that we are doing a lot to promote microphysiological systems. I'm going to talk about a parallel initiative in which we, over the last 8 years, involved far more than 100 people, which is good cell culture practice. It's all about quality of microphysiological systems and cell culture in general. I have to mention some conflicts of interest. Here, especially relevant are my involvements in the microphysiological systems area. So please take everything I'm saying with a grain of salt. You have seen the revolution. We are part of it, which is taking place through 2 workshops, which were organized by Uwe Marx and Adrian Roth, both of them in Berlin. We have been trying to find consensus and promote the acceptance of these methodologies. We were very happy that the 2nd workshop led to this science perspective paper more recently, but I don't need to preach to you about MPS and their aspects. As I said, it is all about quality. And I would like to talk about mainly the first 2 steps, which is good cell culture practice, which is about how to do cell culture and in specific microphysiological systems. I will talk about the starting project of in vitro reporting standards. And ultimately, this is all leading to something which is somehow qualifying and validating these systems for actual use. Irreproduced cellability is a problem. And we don't get to good systems if we are putting bad cells into whatever fancy bioreactor or whatever we're having and call them MPS. And there's a lot of aspects to it. I will be very brief on this. We have to assume that still about 25% of the cell lines in academic use are not what they're supposed to be. It is consistent over time. The last study I know of is from 2015, which found pretty much 25% misidentified. The second point, we still live with mycoplasma infections. Again, something between 15 and 25% is likely infected. This is something which is easy to control, but it's not controlled everywhere and is impacting dramatically on our outcomes. So there's genetic instability. This problem is a bit less with iPSC, but not gone. We have genetic instability, and we have to be careful how long we are propagating our cells, something we have to tackle. This was for me, eye-opening study where we found that 2 vials of cells, which came from the same batch from ATCC, probably one of the best cell culture banks in the world, had dramatic differences in their behavior. And when we took materials, again, from the same batch, without even sawing it, we found dramatic differences in 6 chromosomes within these same vials from the same batch. So there's still some quality control to be done, how we produce our vials. This is another example showing you the problem of these cell lines, the MCF7 cells, again, like in the previous study, which have been given rise to more than 30,000 scientific papers. And in this paper, analysis clearly shows they're not even typical for breast cancer. It is comparing the MCF7 cells with actual materials coming from patients. So it is very, very problematic what we're often using. And this is not only the MCF7, this is the HeLa genome. In the end, it shows it is a Franken cell. It has some chromosomes completely missing. There is reshuffling of essentially everything. It is really dramatic. The MCF7 cells had lost 10% of their genome. About 40% was duplicated up to 40 times. Yeah, so it was really an enormous sack of genetic materials only. Here, another example. These are commercial cell lines. You can pay a lot for this. Supposedly, the only difference is that one gene was inserted, a P450 enzyme here, and you can see they are very different. Complete chromosomes, again, not identifiable in one. This is something where you would interpret your findings by saying, yes, this is with and this is without P450, but it is much more which has changed. So altogether, irreducibility is the problem. Cell culture artifacts. Our use of antibiotics makes our life easy, especially with complex systems like an MPS, but they are producing a lot of effects. This is just the gene expression which is induced by the use of standard antibiotics. We have a situation where we culture normally our cells by like pan-fried eggs, sunny side up. That's how our cell culture looks like. And we are jointly trying to move this to three-dimensional situations, which also give us then finally cell densities, which are tissue-like. So not all our factors being diluted, percentages of the cell-cell contacts which normal tissue has. So there's an enormous advantage, and MPS are for this reason alone a very important step forward. But there's more artifacts in standard cell culture. The sudden change of the environment by changing our media, suddenly replenishing all of the nutrient and oxygen which has been used and removing the waste. This is a situation where a cell has to stay adaptable, and being adaptable is the opposite of the differentiation. It is a very important step that we are introducing homeostatic conditions. And this does not hold for all research. Our own organoids, we are changing the media every second day. It is not by moving to an organ that would have overcome this problem already. It is really important that we consider moving more to homeostatic conditions. And there's many more to mention. We have obviously a problem with bad cells in a device. But the use of primary cells, which are increasingly becoming commercially available, the use of stem cells is helping us at least to get better human cells available. But is then the lack of organ interactions? You know, we have seen it, body on chip type of solutions. That's what is really the future of allowing us not only to have the tissue physiology and cell physiology reflected, but also some of the actual physiology of the body. I believe that the 21st century science requires 21st century cell culture. And this is where all of these MPS come in. This is what it's all about, to really improve the quality. Already in 1996, I started an initiative which is called Good Cell Culture Practice. At the time, organizing a symposium for the German Cell and Tissue Culture Society. This led 1999 to a call on the scientific community to create GCCP, and I had the privilege to lead the task force for ECVAM before I later became the head of ECVAM. And in 2005, we published for the first time a document through the Commission on good cell culture practice, which has been reasonably cited since then. It is talking about what you should know about your model. It's talking about the assurance and quality of all materials and methods you are applying. It is talking about documentation. So what is the minimum documentation necessary if you describe such systems? It talks about hazards. It talks about the laws and regulation and ethical principles. And it talks what should be the education which is advisable to do high quality cell culture work. So these are the 6 principles of GCCP. But 2005 was before even iPSC were described. So it is very meager on stem cell work. It is not at all mentioning anything which is bioengineered microphysiological systems. So in 2015, we held 2 workshops on both sides of the Atlantic. These are the organizations which teamed up at the time. And these 2 workshops produced workshop reports and were the starting point for revision of Good Cell Culture Practice 2.0. Good cell culture, these are the respective reports. Good cell culture practice is not alternative to good laboratory practice. It is to some extent overlapping, but it is meant for those people who are cannot apply good laboratory practice, because good laboratory practice, for example, requires that every person is educated before running the experiment. In an academic lab, this is not the case. We are learning or training our students while they're doing things which we still want to publish. So it's more a minimum standard we are trying to achieve here. Good laboratory practice would require that each experiment has one dedicated workplace. Our hoods are used for tons of projects. We cannot afford a standard of GLP, but we can work in a spirit towards GLP, and this is what good cell culture practice wants to be. It wants to give guidance to journals and funding bodies, and this is what we try to achieve. And finally, in January, we published the new guidance document. The draft had been published exactly a year earlier, and we received 350 comments from more than 100 stakeholders, which were discussed and worked into this by the drafting group, the steering group, which you can see here. So I invite you to look for this document, make use of it. We are entering now a dissemination phase. We are trying to get this to the respective stakeholders. We hope that funding bodies will consider requesting GCCP standards for execution of work, because we feel that is something to promote our work. Interestingly, a parallel development with some overlap in personnel led the OECD to adapt their good laboratory practice guidelines to in vitro. Because good laboratory practice had been developed originally around the animal experiments which were done in OECD. And they call it good in vitro method practices. And interestingly, it is copy pasting a lot from GCCP 1.0, and it's in full annexing the first of the workshop reports we did, the only thing which was published at the time of their publication in 2017. So this is a very important development, and we are continuing to crosstalk with them about adaptation. The second element, which I think of our work, which is not known to most of you, is we are interested in bringing evidence-based medicine to toxicology. We call this evidence-based toxicology. I started this also at the time of the commission. We held the first conference on evidence-based toxicology in 2007. Since 2009, I have the privilege of a chair for evidence-based toxicology at Johns Hopkins, and since 2011, we are hosting the Secretariat for the Evidence-Based Toxicology Collaboration, an initiative which involves the FDA, the EPA, the European Food Safety Authority, and others in the Board of Trustees, and many, many activities. More than 900 people have worked on the evidence-based toxicology collaboration. I'm mentioning this because there's again a tremendous overlap in quality assurance, the desire to come to good data, to good, to high quality things, and come from good data to good conclusions. This was a paper, one of the outputs of this collaboration, which was guidance on assessing the methodological and reporting quality of toxicology relevant studies. And it shows what are the frameworks to assess whether in silico, in vitro, in animal data, human data, epidemiological data are of high quality. So I invite you, because this is something which is critical and identified as one of the needs. We don't really have enough for evaluating in vitro studies. The ToxR tool, which also came out of the evidence-based toxicology movement, was the only one really suitable for qualifying papers. So an ongoing project is the IV-CAT, the in vitro critical appraisal tool. This is again a tool which wants to judge on the basis of a publication, is this good data? Is this a system which is suitable for working? So this is an ongoing process out of the EBTC, which I see very much in parallel to these activities. The second point I would like to make, and this is a project which has started in 2019, where we started with this white paper on good in vitro reporting standards, because it's not only to do the work well, but also to communicate it well to others. This goes beyond documentation, which is internal to the lab, which GCCP covers. It is about what we are doing in order to quality assure our publications. And I believe that a lot of the reproducibility issues we have in science are actually miscommunication issues. We are not talking well enough to each other on how to reproduce an experiment, because we still limit ourselves in the way we are describing the models, and we do not systematically apply reporting standards. This is an example where we developed a template for cell-based toxicological test methods. Similar templates could be developed for others. I think it is really important to set standards here and help a journal to have checklists of, is this system adequately described? Many papers are useless for the scientific community just because they miss important information on what has been done, what has been blinded, which passage of cells has been used and others, the metadata around our experiments. This is where we need to improve and to build on. And you see, from my point of view, this is something where we really have a piece where simple cell models should be quality controlled by GCCP. The microphysiological systems now covered in the 2.0 version. The in vitro standards from OECD come in if this is for regulatory type of testing, but there's reporting standards now to be produced. It is about the quality storing of the publications we do from this. And in the end, this ensures that an information need is actually satisfied. And with this, I would like to end. I cite always or almost always in my presentations John Maynard Keynes, an economist, because he said something very wise, which is the difficulty lies not in the new ideas, but in escaping from the old ones. I believe that for our escape from the old ideas, from the systems of the past, quality assurance will be critical. And I hope that GCCP and GiveRest will be important contributions to help the community. Thanks a lot.